Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello students, welcome to the class. In last class, we have started discussion with the subphases of prophase 1, and we have started with the lactoting and followed by the zygote, and there are few more things to be discussed under zygote phase. In zygote, I told that there is a pairing of homologous chromosomes takes place. That means the chromosomes of the similar kind will come together. They are homologous because the type of genes present in them will be same, but the difference is one is maternal, another one is paternal, I told. And one more thing to be understood here is the pairing is very specific. The pairing of homologous chromosome is very much specific, which means the specific regions of this chromosome will come in contact with or will be in parallel condition with the opposite one. That means if you consider this as one homologous chromosome and there is region A, B, C, D for that matter, and the other homologous chromosome will come in contact with this. That means homologous pairing will take place, right? This pairing will be like the region A will be parallel to A and region B will be parallel to B, C will be parallel to C and D will be parallel to D. I taken this alphabetical denotion in order to understand, but you have to understand that these are genes. The genes which are present on one homologous chromosome will be parallel or will be in a close contact with the same chromosome, same type of chromosome, not of different kind. Like here D will come, no. Here C will come, no. Where there is B, there will be B. Where there is C, where there will be C. That means the type of genes, the regions on one chromosome, the specific regions on one homologous chromosome will stay in contact with the same regions of the opposite homologous chromosome. That is, the homologous pairing is very much specific. And this pairing of homologous chromosome may be of different kind depending on which region will come in contact at first. That is, based on that we can classify the types of pairing into three types. One is procentric pairing. Procentric pairing. Procentric pairing means here is one chromosome, right? So this is, and here is the other, the opposite one, that is homologous. Here the pairing of chromosome will starts at centromere. The pairing will start at centromere. Here the pairing will start and proceeds towards the end. The regions at the centromere will stay together and it will be followed by the arms. That is called procentric pairing. And the second type is proterminal pairing. Proterminal pairing, where first the edges, the ends of the chromatids will come in contact and it will be followed towards the centromere or towards the center. That is called proterminal. That is, the pairing will start at edges or ends and it will be proceeded towards the centromere. That is called proterminal. And the other type is random. Third type. Random pairing, or there is no specific thing, it will start at centromere or it will start at terminal. It may starts anyway. So that is called random pairing. So these are the things to be discussed under the zygote phase in addition to whatever we have discussed from the last class. And after the pairing of homologous chromosomes, that is, the similar kind of chromosome will stay together. We have written that. That is, I will write a phase where the cell is in zygote. Here there is a nuclear membrane as such it is present, and the centrioles, they just started moving towards the opposite poles. They have not yet moved to the ends, and there is a pairing of homologous chromosomes like this. Here is one and here is another, and this is homologous pair, and here is one more pair, one more homologous pair. Since we have taken 4 chromosomes initially, I am having a track of 4 chromosomes. And I told the chromosomes are not having 4 chromatids. Sorry, in a dyad, they will not be having 4 chromatids in the zygote, because the chromosomes are the thin thread-like structures in zygote. This is zygote. Now, which I discussed, what happens in the next phase, that is the third phase. This is the second phase, right? And the third phase of prophase 1 is pachytene. And the pachytene, pack here refers to, the thick, and tene refers to threads, which means there is still more condensation of chromatin takes place. There is still more condensation of chromatin, hence the chromosomes appear very thick. Compared to the zygote. The chromosomes will be very thick, hence we are giving the name pachytene. Pachy here refers to the thick. That means because of the still more condensation of the chromosomes or the chromatin network, the chromosomes appear very thick. And there is a process called crossing over. Crossing over takes place in the pachytene. Here, to show that here is a cell in the pachytene, no doubt the nuclear membrane will be present as such. There is no change for the nuclear membrane, as well the centrioles. And here, during the pachytene, the cells, chromosomes will be very thick like this, and they are present in close contact, right? Here, they overlap on each other like this. So, I'll just draw this with a thick one and one other transparent, so that it will be easier for you to understand. Here is the one pair where it has undergone crossover. So here is the place where there is a crossing over. Here is crossing over. Same will happens for the other set also. And here also I will be writing one transparent. Both are homologous, right? So I will be writing in the same color, but one transparent and one opaque. So when you are studying also make like this, so it will be easier for you to differentiate the things. So like this, which means during the packaging phase, the two non-sister chromatids of a homologous pair, non-sister chromatids of a homologous pair will overlap and exchange the part of it with the other. And this process is called as crossing over or it is called recombination. The process is called recombination. It is just to understand. Here is one chromosome and here is the homologous chromosome, the other. These two are called non-sister chromatids. I already discussed it. The chromatids of a homologous set, that is the neighboring chromosomes, are called the non-sister chromatids. And the chromatids of a same chromosome are called sister chromatids. Here, in packaging phase, the non-sister chromatids will overlap on each other. Just imagine these two are the chromosomes, chromatids of a homologous chromosome. They just overlap like this. After overlapping, what happens? A part of this will get exchanged with this. So, part of it will get exchanged with this. So, this process is called as crossing over or recombination. There is a combination already, but that combination is rearranging. The part of this chromosome will transfer here and the part of this will be transferred here, and the process is called recombination. It will just appear like this to show you. I write the recombined chromosome here. This is after recombination. Actually, after recombination, the chromosomes are not visible separate in the packaging. The process will happen in the next phases. For better understanding, I am writing this here. So here is the homologous chromosome. What happens here is after recombination, part of this chromosome will be here. Part of this chromosome will be here and the part of will be here, like this. So wherever there is a crossover, wherever there is a crossover, there will be an exchange. So this process is called as recombination. For this recombination, anyhow, the chromatids are made up of DNA, right? So DNA should be cut here. DNA present here will be cut and it will be joined here. For cutting and rejoining, the recombination or the crossing over process is enzyme mediated. There is a requirement of enzyme. The process of recombination is enzyme mediated. That is, for recombining the chromosomes, there is a requirement of enzyme. And the enzyme involved here is recombinase. In the presence of recombinase enzyme, the crossing over will take place, and this recombinase is having two functions. One is cutting the DNA or cutting the chromatids. In higher classes, you will study how it will cut the DNA exactly, but for here I will be just giving it as chromatids. Cutting the DNA, or we can call it as nicking, and joining the chromatids. First it should cut the chromatids, later it should join the chromatids, and joining is called annealing. Nicking and annealing. So recombinase enzyme is having the two functions. In the presence of recombinase enzyme, the crossing over will take place. And remember, this is a very long process for activating this enzyme. After that, it should act on the DNA, then it should cut, it should rejoin. For that, it requires some time. Hence, compared to the previous two phases, that is leptotin and zygote, the packaging is long-lived. That means duration of packaging will be more compared to the leptotin and zygote. So these are the things to be remembered in the packaging. The fourth phase in the prophase 1 is diplotin, and here diplo refers to 2 and tin refers to threads, which means that the two homologous chromosomes which were stayed together in the zygote and the packaging are getting separated as a 2 threads. So that's why we are referring the term diplotin. And here in diplotin, what happens? There is a dissolution of synaptonymal complex. Synaptonymal complex. We have discussed in the previous class, the synaptonymal complex is an arrangement of specific proteins due to which the two homologous chromosomes are staying together. But when the cell enters into diplotin, this synaptonymal complex will become unstable and it is getting dissoluted. So because of the dissolution of synaptonymal complex, the homologous chromosomes starts separating except at the crossover. Wherever there is a crossover, apart from that site, the chromosomes are getting separated. I will show it in a diagram. So here is a cell in a diplotin. Here is the nuclear membrane as such, it will be present, and the centrioles. And here what happens is, the chromosomes which have got crossed over are getting separated apart from each other like this. They are getting separated, distance will be developed between them, but the crossover site will be as such. Here is a crossover, it is like this. And here one more set I will be writing here. Here also the same thing. So the two homologous chromosomes are separating in this direction. They are getting separated, but at the site of crossover, here is the site of crossover. At the site of crossover, the chromosomes will be staying together. Here is the crossover is not dissoluted yet. That means at the site of crossover, the chromosomes are staying together. Apart from that, they are separating. So, because of this process, there is a formation of X-shaped structure. When we enlarge it, it will just appear like this. Only this part I am enlarging here. Only this part. So, when we enlarge it, it will just appear like this. So, can you say how it is appearing now? It is just appearing like a X shape. So, because of the separation of chromosomes apart from each other, except at the site of crossover, there is a formation of X-shaped structure which is called chiasmata. It is a characteristic feature of diploting, and generally in all organisms, there is a formation of chiasmata. Generally in all eukaryotic organisms, there is a formation of chiasmata, but there are some organisms where the diploting will be achiasmatic. For example, in case of some dipterians, for example, Drosophila. In case of Drosophila, housefly, there is no formation of chiasmata, achiasmatic. So it will be important for competitive exams. And in some organisms, the position of the chiasmata may change. Generally, it will happen at the terminal, but in some other organisms, the position of the chiasmata may change. So, based on that, there are two types of chiasmata. One is terminal chiasmata, which means whatever I shown here, the chiasmata which is formed at the end of chromatids. Is called the terminal chiasmata, and another type is interstitial. Interstitial means it may happen anywhere in the chromatid. That is called interstitial chiasmata. So these are the events which are taking place in the diploting, and remember, the chromosomes have not separated completely they have not separated. They are together at the site of chiasmata, but they started moving towards the opposite, they started moving apart. So that is the event which is taking place in the diploting. And the next phase is the last phase, the fifth one, diathenesis. Final stage in the prophase 1 is diathenesis, where here dia refers to across and kinesis refers to movement. That means the chromosomes are moved apart, across, they have moved completely. So we are referring the term the diathenesis, and it is found to be the final stage of prophase 1, and the chromosomes are still condensed. And appear very thick. And you should remember that after diathenesis is the metaphase 1, and we have discussed from the mitosis that only in metaphase we can see the complete structure of chromosome. So that you should keep in mind. And in all these phases, that is from leptotene to the diakinesis, there is a condensation of chromosome is getting more and more from phases to phases, and it is terminalizing or it is completing at the diakinesis. So because of that, at the end of diakinesis, it is possible to see the complete structure of chromosomes because of still more condensation. And here the chiasmata will be separated. That is, the chromosomes, the homologous chromosomes are staying together at the site of chiasmata, right? That chiasmata is also getting separated at the diakinesis, and the process is called terminalization. That is, because of still more condensation of the chromosomes, the chiasmata will get separated. At the site of chiasmata, there is a separation. As a result, the two homologous chromosomes are now completely separated, and the process is called as terminalization. And after terminalization, there is a dissolution of nuclear membrane. This is the end of prophase, right? At the end of prophase, there is no nuclear membrane. That is, the nuclear membrane is absent. That is, it is instead of absent, I will write it as the nuclear membrane will get disintegrated. The nuclear membrane will disintegrate into vesicles. And nucleolus will disappear. Since there is a dissolution of nuclear membrane, the separated chromosomes are suspended in the cytoplasm. The chromosomes are suspended in cytoplasm. The chromosomes are suspended in the cytoplasm, and there is a centrioles which have reached the poles. Centrioles have reached the poles. They have reached the opposite poles, and one will be here and one will be here, and there is a formation of complete spindle apparatus. Like this, complete spindle apparatus will be present, and there is a distribution of chromosomes in the cell cytoplasm. And remember, when you are writing the chromosomes, they are undergone recombination. That you should keep in mind. So here, this is one set, crossover region I am showing here. And one more set. Don't think they are staying together. They are suspended in the cytoplasm, right? So we can write any way. So this is how the chromosomes will appear here. So now this is the end of diakinesis, where there is a complete separation of the homologous chromosomes, and there is a dissolution of chiasmata. As well, there is a disintegration of nuclear membrane into vesicles. As a result, the chromosomes are now free in the cytoplasm, distributed here and there. Okay, so this is about the diakinesis, last phase. After this, the cell will enter into the next phase of division, that is the metaphase. We shall continue that in the next class.